Hello everyone, in this video I will demonstrate how we used ASHOP to investigate cell developmental subgroups within the DUX4-rearranged BCP-ALL. We have about 6,000 cases in this program. To focus on only the DUX4 subtype, we can navigate to the filter tab to define a custom filter for DUX4 subtype. Select molecular subtype and check only DUX4. Clicking apply and then only the DUX4 samples will be retained for further analysis. From the transcriptome UMAP under sample scatter plot, we identified two distinct gene expression clusters within the DUX4 rearranged BCP-ALL. We could zoom in from here and reposition the plot by dragging it. To compare the two clusters, we could lasso the two clusters and save them as new sample groups. The new sample groups created will be shown in this Groups tab. Once the two DUX4 clusters are saved as sample groups, we can create a custom variable using them by clicking this button. A new custom variable will be created, and clicking it will be able to use various tools to compare the two groups. We first conducted the differential expression analysis to identify genes that are differentially expressed between the two clusters. We found that B-cell differentiation genes such as EBF1, MME, and RANK1 were highly expressed in one cluster, and lymphoid progenitor and myeloid and inflammation genes were highly expressed in the second cluster. In addition, GSEA analysis of DE results revealed significant enrichment of inflammatory pathways. These findings agree with the recently identified early multipotent and committed subgroups in DUX4 rearranged BCP-ALL. So we performed a clustering analysis using multipotency score and developmental state abundance, as previously described, to separate the DUX4 subtype samples into early multipotent and committed subgroups. And we put these subgroups in a term named molecular subgroups in the data dictionary. We next created two new sample groups. So delete the two old groups and delete the old custom variable. Add group, select molecular subgroup, check DUX4 early multipotent, and another group, select molecular subgroup and check for DUX4 committed, and give them new name. So DUX4 early multipotent and DUX4 committed. And we create a new custom variable using the two groups. Clicking the new custom variable, we'll be able to compare the two groups. We first compared the survival outcome of the two molecular subgroups. So clicking the custom variable and launch compare survival, select overall survival, and we do the same for the event-free survival. As shown here, the early multipotent subgroup exhibited inferior event-free survival, but not overall survival, than the committed subgroup. To compare the NRAS mutation status between the two subgroups, we created a lollipop plot. So from the custom variable, select compare mutations and type NRAS. Using our lollipop plot, we found that NRAS mutations enriched in the early multipotent subgroup and is rare in the committed cases. These NRAS mutations were predominantly glycine at position 12 and 13 hotspot missense mutations, with position 13 mutations exclusively found in the early multipotent subgroup. To further compare the NRAS mutation status between the two subgroups, we overlaid NRAS mutation status on UMAP. So from the transcriptome UMAP, clicking the color term, replace, select mutation tab, and type NRAS. Edit and no sample grouping. We can zoom in from here and reposition the plot by dragging it. We are not interested in CINV, so we do not show the CINV for now. By overlaying NRAS mutation status on UMAP, we identified that 3 out of the 4 NRAS mutated samples of the committed subgroup localized close to the early multipotent cluster, suggesting that NRAS mutation is primarily associated with the early multipotent classification. That brings us to the end of this quick tutorial. For the full tutorial, please visit proteinpaint.sanju.org/ashop/tutorial. And if you have any questions or suggestions, please contact us through proteinpaint and sanju.org. Thank you for watching.